Hi, everybody. We're going to be doing week 3 review. And so this will be covering chapters. I'm sorry, I'm trying to minimize this. Chapters 2 and 4. Here we go. Chapter 2 is covering the chemistry of life, and chapter 4 is covering bacteria and archaea. So we're going to go over that content today. Starting with chapter 2, these are our key concepts for chapter 2, the types of bonds, starting with covalent bonds. In covalent bonds, basically what we're talking about is sharing of electrons between atoms, whereas ionic bonds are the transfer or donation, where quite literally an atom is giving the electron to another atom. And this stuff depends very highly on understanding like what an atom is and how an atom works. Remember that an atom, so bonds are things that happen involving atoms interacting with one another. So between two atoms, an atom again being consisting of neutrons and protons, neutrons being neutral in charge, protons being positive in charge in the nucleus of the atom, and then surrounded by electrons that orbit that nucleus, okay? And how many protons determines how many electrons you should have in an element. And basically based on how many protons you have determines what element you are, and then you should have an equal number of electrons in a typical atom. Then there are different levels of the electrons and what they exist in, 2 and then 8 electrons want to be in the second one, and then 8 in the next one, and so on and so forth. Basically determines how many electrons that atom wants to have in its orbital. And the atom is going to be happier with, that set number of electrons in that orbital. So some atoms might want to give up an electron, and some atoms might want to donate an electron or accept an electron, depending on how many electrons they have in their orbitals. And that is what basically causes molecular bonds to be formed. So if we can share electrons to fill our orbitals, that's where our covalent bonds are coming into play. But if we can donate that electron completely, and now we're happy without that electron completely, and the other atom is happy to have that electron completely, that is where we see ionic bonds. And so atoms like sodium chloride, this would be a molecule, right? A molecule like sodium chloride, where you have an atom of sodium and an atom of chlorine, where that sodium becomes positively charged and the chlorine is negatively charged. This is because the sodium gave up. Let me write that clearer. That sodium literally gave up an electron. And that chlorine literally accepted or stole or gained that electron. And now they're both happy that way. And typically, as molecules will do, they're fine to hang out with each other because we've got a positive charge and a negative charge, and they'll generally just hang out with each other as a result of that. And that is what the ionic bond is. And then whenever we introduce those into water, where water, again, we have H. The oxygen in a water molecule, here, this line represents a covalent bond where oxygen is sharing with hydrogen, but it is uneven sharing because oxygen is electronegative. It tends to have a negative polarity because it's hogging the negative electron the most, whereas the hydrogens have a positive polarity because they spend less time with the electron. And now those polar positive hydrogens can interact with the chlorine through what kind of bond would that be? There's be an attractive force between a polar positive hydrogen and something else that is either negative or polar. This would be a hydrogen bond. Okay, no electron exchange, just attractive forces with a positive polar hydrogen there. And then this sodium is positive and could interact well with the negative polarity oxygen. Okay, and so these attractive forces can then pull apart those ions that were ionically bonded and then ionize those ions into a solution with water, where they can start interacting with the water molecules because of the polarity of water. And this is what makes water the solvent of life. Its polarity, the tendency of the hydrogens to have a positive polarity and the tendency of the oxygens to have a negative polarity there. All right. So that's the gist of how water is going to be dissolving ionic compounds as well as polar compounds. And it's why water doesn't mix well with nonpolar things. So what does nonpolar really mean? It means it doesn't have any charge. There's no electronegativity. Things are sharing their atoms with one another very equally. Nobody has any charge as any result of that. So that does not mix well with water. Things like hydrocarbons, like our lipids and waxes and things like that. Okay, so that's the gist of covalent bonds and ionic bonds, and also hydrogen bonds, which are just attractions between a positive polar H and something else that's usually either negatively charged or negative polarity. Okay. All right. Next, we talked about organic molecules. Organic molecules must have a carbon that is bound to a hydrogen. It is not enough to have carbon in it. For example, carbon dioxide. There's no hydrogen bound to the carbon. It is not organic. Must have hydrogens bound to the carbons for them to be organic. So things like sugars and stuff like that, we have hydrogens bound to the carbons and all that. Get into the sugars and all that in a little bit when we talk about our biological macromolecules. All right. So we did introduce this concept of polarity, and polarity basically dictates whether you are hydrophobic or hydrophilic. If you are hydrophobic, remember phobic equals like fear, basically. You have a phobia of something, you're afraid of something, right? And philic means love. The city of brotherly love, Philadelphia, okay? So in this case, hydrophobic, fearing water, and hydrophilic, loving water. All right. And then these terms basically have to deal with the polarity. If you're polar, you're hydrophilic. You love water because you can interact with those polar water molecules very well. Okay. If you are hydrophobic, you are non-polar. You don't really have any way to interact with those polar hydrogens or polar oxygens or anything like that. So you don't mix well with it. So a lot of our fats are very non-polar hydrocarbons, which are not going to have any sort of charge or polarity with them. They don't mix well with water as a result of that. So there are some molecules like a phospholipid, like you have PO4, which is a negatively charged, and then a long hydrocarbon chain tail. That the head, because it's actually charged, is hydrophilic, and the tail, being hydrocarbon, is nonpolar, which is hydrophobic. When you've got both hydrophilic and hydrophobic to something, the term for that is amphipathic. Amphi always means both. All right. Next, we have the pH scale. So let me clear my screen. Whoa. What I meant to do. Just, it's not wanting to. There we go. Okay. Wish there's just a button that would let me clear the whole screen. There probably is, and I'm just not smart enough for it. All right, so next we're going to talk about the pH scale. The pH scale depends very heavily on hydrogen ion concentration, so those H pluses. Whoops. All right, so literally the pH scale. pH means the negative logarithm of the concentration of the hydrogen ions. I'm not going to make you do math with it, but what this means is we know our pH scale goes from 0 up to 14, and we know 7 is in the middle or neutral. And if you're less than 7, you're going acidic. And if you're greater than 7, we're going to basic or alkaline. Okay. This is the gist of the pH scale here. But 7 is based off of water. And this is all based off of water. And what's really going on here, if we say 7 is neutral because H2O is going to make equal amounts of H pluses and OH negatives. When those are equal, that is 7. Okay, and this is important also because what the negative log of the hydrogen concentration really means, it means that 10 to the negative, whatever your pH is the actual concentration of the hydrogen ions. So this is important because which number is bigger, 0.1 or 0.0001? 0.1 would be a pH of, you only have to move it one place, 1, 1, 2, 3, 4, and this one's a pH of 4. That's literally what it means. That's literally what this is. So this all is in respect to that concentration and the ratio, basically, of the hydrogen ions in a solution. A pH of 0 means that your hydrogen ion concentration, basically what it means is that your H plus concentration equals 100%. It's a ratio of 1. Okay, and that means that it's 100% hydrogen ion. You can't have more than that. So you're never going to have more than 100% hydrogen ion concentration, which is why 0 is where it ends at. All right. And so this is all based around water and that the H pluses and the OHs are equal. So really what you're talking about when the pH itself goes lower, when you approach 0, you're having more hydrogen ions approaching 100%, hopefully never getting there. Or as you approach 14, you're approaching 0.0000014 decimal places over, which is as close as we're going to get out there. And that just means you have less and less and less and less hydrogen ions in comparison to water. But a lot of our comparisons that we were doing in biology aren't going to necessarily be in comparison to water. Sometimes it is, but sometimes I might ask you to compare it to other systems. And that might be blood, for example. For blood in humans, this is humans, right? The pH is 7.35 to 7.45. And if you deviate outside of that, you've got problems. So why would you have problems? Because the other macromolecules in the cells rely on this pH in order to hold their hydrogen bonds together. So if they're relying on those, H pluses, these dipoles, polarity of positive H's, being attracted to oxygens that have a negative polarity to attach to whatever. That hydrogen bond is depending on that attraction to hold the protein together or to hold your actual DNA ladder together or whatever. That can't happen the way that it's supposed to if you have too many free H pluses hanging around. Because instead, the oxygen will interact with that. And now you've lost your hydrogen bonds. So this is what comes at risk first. Then things like salt bridges, which are ionic attractions, things like even disulfide bonds can get disrupted if you have too many H pluses that are around. And so these things can actually significantly alter the structure of our macromolecules if the H pluses are too high or too low, basically interfering with the ability of our macromolecules to form these other bonds that they rely on for their structure. And so it's really key in biology that we understand the effect of our pH on our systems there. Okay. Okay, there's just a button I could click to remove all of my drawings, but there's not really. It's fine. Okay. No. That helped. Okay. So anyways, that's pH. And that's just a pH. So keep always in mind that we're often making comparisons of a pH to another pH and whether or not they're more acidic or less acidic. But not just that. How many hydrogen ions? Is there more hydrogen ions or less hydrogen ions, comparatively speaking? The lower you go, the more acidic, but that means the lower you go, the more hydrogen ions as well. All right, so that's pH. So we're talking about pH in respect to our biological macromolecules. Our macromolecules, our macromolecules are basically carbohydrates. These are going to be our saccharides. A lot of these end in ose. It's not just going to be glucose, but think about ribose. Think of deoxyribose. Those are really important ones too. If they end in ose, it's typically a sugar. Even if we talk about cellulose, that's making up the wall of plants, for example. So those oses, a lot of those are basically sugars: lactose, fructose, sucrose, glucose. A lot of them are sugars. Okay, so sugars fall into that category. The subunit of those are going to be our monosaccharides. Okay, so that's our first one. Our second one is going to be our lipids. This includes, there's not really a subunit for this one. We might say the fatty acids or the hydrocarbon tails even, but there's not really a strict subunit for this, but this includes groups that include our fatty acids. This includes waxes. This includes sterols like cholesterol. And these are often precursors for steroids. Okay, so related in that manner. And triglycerides. Which triglycerides are just a glycerol molecule, which is a 3-carbon backbone, off of which 3 fatty acid chains are attached to. And that's really all that those are. And we can categorize them even further based on whether they have double bonds or if they're all just single bonded with carbon, which would be saturated and that sort of stuff. So those are our lipids, probably really important ones we will introduce as we go through them. But things like cholesterol obviously is a supportive sterol lipid in our membranes. And a very important lipid would be phospholipids, which are an amphipathic version of a lipid that's important for our membranes. They create that lipid bilayer that makes up our membranes, makes up the membranes of eukaryotes and bacterial cells. Okay, so really important one there. The third one will be our nucleic acids. This would be DNA and RNA. We'll learn more about the specifics of our DNA and RNA, as well as ATP, adenosine triphosphate. With the base of all of these are all made of nucleotides. And what is a nucleotide? I'm so glad you asked. A nucleotide is basically a 5-carbon sugar. Okay, I drew it probably as sloppily as I probably could imagine it being. But here's our 5-carbon sugar that is attached to a phosphate, which is a PO4, with a negative charge, and it is attached to a nitrogenous base. That nitrogenous base is usually a ring-like structure. It is a purine or a pyrimidine, and for DNA is A, T, C, or G. For RNA is A, U, C, or G. We will come back to this in Unit 2 in Chapter 6, but we're just reminding you about the general structure of DNA and RNA. Remember that in DNA, you have your sugar phosphate backbone. And then off of each of these sugars, you have a nitrogenous base. And again, if that nitrogenous base was A, it will be hydrogen bonded to a T on the other side. And if it was a C, it will be hydrogen bonded to a G on the other side. Quite literally, and repeat over and over again. That's what's really happening on that DNA ladder when you unwind that and really look at the molecular structure of it. And that, a sugar phosphate with a nitrogenous base, that is a nucleotide. So that's the gist of the nucleotides, okay? That's the general understanding. These guys are really important. They make up your genetic code, and they're also like our intermediates. When you take your genetic code and then turn it into protein products that the genetic code codes for, your DNA is your blueprint. Your RNA is like your working tools to build things. And then the actual product is the protein that we'll get to. We'll talk about that in chapter 6. It's a unit 2 thing. Transcription and translation and all that stuff. Just reminding you about that coming up. All right. So anyways, this is taking too long. All right. So lastly, the protein level. So our last macromolecule group is the proteins. Proteins are made up of amino acids. And in fact, that's the first level because there's 4 levels of structure. First is called the primary. That's just the amino acid sequence. And what's an amino acid? I know. Amino acid basically is, and let's draw one out over here. Okay, we have a central or alpha carbon that has a hydrogen. Remember, all carbons always bind to have 4 bonds. It's bound to another carbon, and it's also bound to a nitrogen. The carbon that it's bound to is a carboxylic acid, OH. And that means that that H can potentially become a H plus. That's what acids can do, right? They can donate H pluses. So this is the acid side of the amino acid. The other side is the amine, go figure, which is an NH2 group. So there's an amine and there's an acid, and that's why it's an amino acid. Okay, so it's really all it takes. But we still needed 4 bonds on that central alpha carbon. And that 4th bond binds to the R group. Whatever that R group is, then determines what amino acid you are. Are you alanine, phenylalanine, tyrosine, threonine, glutamate, whatever? Okay. So there are 20 different amino acids generally that we see in biology. And so we'll figure out in chapter 6 how we code that in your DNA. But basically, of those 20 different amino acids, when you build your protein, you have methionine, alanine, cysteine, glutamate, asparagine, whatever you have for your amino acid code, that's your primary sequence. So I think of it as building your ribbon. Your amino acids build the ribbon. Your secondary structure is how the neighboring R groups interact with one another. Neighbor R's interact. And this causes certain levels of structures and characteristic things. They could be just random interactions with random folds kind of here or there. But one of the structures that we see developing just from the neighboring groups, and remember, if they like each other, they want to be near each other. If they don't like each other, if we've got a hydrophobic and a hydrophilic next to each other, they don't want to be near each other. So that will cause the R groups of those amino acids to kind of rotate around that kind of backbone, amino acid backbone, and kind of orient based on if they're attracted or if they push each other away or whatever. And this creates our initial structures. It could be random, or it could create these initial kind of predictable structures like an alpha helix or a beta pleated sheet. Alpha helix is whenever that amino acid sequence becomes a helix. A pleated sheet is when you have a straightaway and then something happens that causes it to turn back, and then you have another straightaway, and then something happens where it turns back, and you have a straightaway, etc. And this is a beta pleated sheet. It creates these sheets of back and forth straightaways. Okay, so these are what we see being caused by the immediate neighboring R groups interacting with one another. So I consider this as being like almost like if you had your ribbon and now you've curled parts of it first. Next, we tie the bow. So you could create a whole bunch of, it's tied like this, and then it's tied like this, and whatever. This is usually due to R groups across the structure interacting. So what's that whole structure called? It's called a polypeptide. So now we've actually like made our original, our kind of final polypeptide structure. This will not just be from neighboring R groups, but R groups across the whole polypeptide now interacting. And this will be whenever we create things like salt bridges between ionic amino acids. This could be hydrogen bonds across the way, or this could be disulfide bonds, which are covalent between like sulfur groups, all of that stuff. So this is what's causing that polypeptide to get its overall shape. So within your bow, you might have a curled little tail from your alpha helix, and then you might have some straightaways that zigzag like this. Wow, that made a heart just all on its own, didn't it? And then you might have another straightaway that goes like that, and then a tail that curls, for example. And that would be your tertiary structure. And maybe these guys are disulfide bonds. Who knows? All right. And that could be your whole structure. But there's one more level to our protein structure, and that is the quaternary. This is when you have multiple polypeptides that work together. So this means you've made that bow, and then another bow comes together, and maybe it looks like something else, or it could be very similar. But now we're bringing multiple polypeptides together to make the bigger bow. When you're wrapping your package, you have to make your bow, you'll curl it, then you'll tie it, and then you maybe put multiple bows together to make one big old, big fancy bow. And now it's functional. So that's the quaternary level. So that is the gist of our chemistry review from chapter 2. All right, moving on to chapter 4. Chapter 4, we have a lot of major concepts, many of which are new ideologies for us, learning about bacteria and stuff. So this is chapter 4 covers bacteria and archaea with a heavy lean on bacteria. Very little mention of archaea because the archaea are not as medically relevant. We don't spend a lot of time talking about them. Most of the archaea live in extreme environments. And so we don't see them a lot in medical settings. So that's why we don't talk about them a lot, but that is something to remember about them. They are extremophiles, and that means they live in extreme environments like boiling water temperatures or like salt lakes and things like that. They live in areas where other microbes can't. And so we don't see them a lot in medicine. There are a couple that we will talk about in later chapters that are medically relevant, but it's not a big deal for us right now, okay? So bacteria is mostly what we're going to talk about. Okay, so we learn in lab 2. If you are my Tuesday, Thursday people, then we did that today. If you're my Monday, Wednesday people, then we're going to be doing that later. But we learned in lab 2 about our cell shapes and arrangements, and that bacterial cells generally can be found in certain shapes and arrangements. The major shapes that we see the cells in are going to be round or rod, or we might have comma shaped. And that can kind of vary on how the comma is shaped, or we might have kind of spiral shaped and whatever. The round cells, or we might have ones that have no defined shape, or have a variety of shapes. The round cells are called coccus is singular, cocci is multiple. And again, I'm not going to be super picky about that on the test. If I say, what is a round cell shape called? And you put cocci, I'm going to count full credit on it, okay? I just want to be familiar with the terms. All right, so rod shape is called a bacillus. So multiple is bacilli. Comma shape is called a vibrio. Spiral shape, it depends on how tight the spiral is or how wound it is and how it has, but it's either spirochete or spirillum. Has spiral in it, basically. And if you don't really have a shape or you have a lot of different shapes that you can be in, then we typically call that pleomorphic. Pleo means many. Morph means shape. Okay. So those are our general shapes that our cells can be in. For our arrangements, this could be bunches, irregular bunches, kind of like grape-like clusters. This is called staphylo. Okay. You could be in chains. And this could be, if you are a cocci, it could be chains like this, or if you're in bacilli, they could be end to end like this. These are called strepto. They can be very, very long chains. And typically, if we are a staphyloformer, you might see some chains coming off, but if you also have groups around them, then you're generally staphylo. If you're strepto, you're only going to be in chains. Only chains if you're strepto, okay? So you can also see that when we look at our bacteria in the lab, that there can be different combinations of these arrangements and how they're set up. So just keep that in mind that you might see both strepto and staphylo. But overall, what would I call it? I'd call it staphylo because if you're true strepto, you're only going to have strepto. So you can even see in a lot of our strepto, we'll often have diplo, which is pairs and stuff like that. So keep that in mind. And there's one other important arrangement to be aware of that I definitely test on. These are our major three, where if you have bacilli, they can be up like this, like a fence. This is called palisades. There are other ones that are discussed in the chapter that I won't ever test you on. So these are our general shapes and arrangements that we have with our, you can also have no arrangement. Sometimes if your cells are just randomly all over the place, and you're not really seeing any definable arrangement, then you might put no arrangement for that, and that would be acceptable. Sometimes this is a little bit subjective, but other times it might be a little bit more obvious. We'll learn more about that in the lab as we're learning about our microbes and their cell shapes and their arrangements as we go through a lot of this stuff in preparation for the lab exams and etc. So that's the gist of the cell shapes and arrangements. All right, so moving on, talking about the structures of our cells now. I'm actually going to start with the general structure of the cell. Now, all of our bacterial cells have an inner membrane called a cytoplasmic membrane. All right, so there's our cytoplasmic or inner membrane. And then many of our cells also have a cell wall. Let's make him orange. Let's make him. If you look like this, we have an inner membrane and a really thick cell wall. Some of our cells have an inner membrane and a little thin cell wall. So this is our cell wall. It's made of something called peptidoglycan. In our top picture, we have a really thick one. In our bottom picture, it's a thin one. Both are made of peptidoglycan. And what is peptidoglycan? It is in the name. Okay. So what peptidoglycan is, it is peptides, peptido. What are peptides? They're amino acids that are linked together, right? So again, amino acids that are linked together. And how I might draw this, let's draw it out to the side over here. Amino acids are linked together. And they'll act as bridges between, let's do a different color, sugars. Okay, 6 carbon sugars. And these sugars, you don't need to know what they are, but for those of you who are interested and like to know all the things, they are NAG and NAM. N-acetylglucosamine or N-acetylmuramic acid. So those sugars are covalently bonded together, and then we create these chains of NAG and NAM that go on forever, and NAG and NAM that go on forever, etc. And they are cross-linked by these peptide bridges. So peptide bridges, glycan, glyco, sugars. Okay, so sugar chains cross-linked by amino acids. In our thick wall, guys, we are many, many layers thick of those chains that are cross-linked by amino acids. In our thin guys, you're only one or two. This can be like 30 to 100 times. And this is like one or two, maybe 3 times. Okay. And that is important because the guys that only have that thin layer of a wall, it's not giving a ton of stability to their cells. So they'll also have an outer membrane. Whereas the guys that have the thick wall, they don't need an outer membrane. But because they have such a thick wall, they also need stabilizers. If they have a thick wall and they need stabilizers, those stabilizers are lipoteichoic and teichoic acids. For our guys that have the outer membrane, that membrane has special molecules on it. Those are called LPS, lipopolysaccharide. Lipo, fats, polysaccharide, many sugars. Okay, so it's sugar, fats, and LPS is also known as endotoxin. A toxin is anything that can cause damage to our cells. So this is a molecule that's just part of this outer membrane. And you're either bacteria that has a thick wall, or you're a bacteria that has a thin wall and an outer membrane. And there are some deviations from these, but generally speaking, those are our biggest groups: thick wall or thin wall outer membrane. Okay, and as it turns out, thin wall outer membrane, these are our gram negatives. I'm write it real big and thick because we're going to talk about it over and over again. Okay, and the guy that has the thick wall is the gram positive. So gram positives have lipoteichoic and teichoic acids to help maintain their thick peptidoglycan wall. The gram negatives have a thin wall. They don't need lipoteichoic acid to maintain it, but they do need an outer membrane to help support them. Okay, so this is the gist of how they're set up. They don't really have any structures inside of their cells other than ribosomes, which are just kind of functional RNA structures. And their DNA and their RNA, those little like molecules, but there's no mitochondria. There's no endoplasmic reticulum. They still make energy for the cell, but it just happens in their cytoplasm, which is the goo inside of the cell. So they still have structures in their cell, they're just not membrane-bound compartments, and they still have functions in the cells, but it's just not in membrane-bound compartments. They also have one DNA chromosome, and we'll learn a little bit more about their chromosomes in chapter 6. But they have one big circle DNA chromosome. It's still double-stranded DNA, helix DNA. It's just in one big old circle, which is different than the eukaryotes, okay? So it's the general structure of these cells, and it gets us out of the way of learning about our gram-positive versus our gram-negative. Some bacteria are motile. Oh, first, before I go on to that, I want to talk about the capsules. Some of these guys might also have a goo on their outside. Let's just say it surrounds them or whatever. And that goo that's on their outside, it could be the capsule. Capsules are usually made of sugars, and they are sticky, kind of like honey. Think of it as being like a honey coating. And if they are loosely attached sugary coating, we call those a slime layer. And if they're densely associated, very tightly wound around them, sugar layer, then that's a capsule. Both of the sugar layers the capsules and the slime layers, they fall into this category called a glycocalyx. These guys are pretty useful for adhering to surfaces. Obviously, if you've got a sugary, sticky coating like honey on your outside, it helps with sticking to surfaces. It also makes it harder for white blood cells to eat them up. That process of our white blood cells eating them up is something that we would call phagocytosis. So having that capsule prevents phagocytosis by our white blood cells. Again, phago means eat, cyto is cell. Okay, so cell eating. So it prevents our white blood cells from eating them up. Okay. Sorry, making my pen smaller. All right, so it also helps them stick to surfaces, so they can adhere to surfaces easier, and often like they can stick even to like the surface of your teeth. And then whenever they do that, they can stick onto your teeth, and other bacteria can stick onto them, and then more bacteria can stick onto them, and so on and so forth, and create their own little kind of happy film in that stickiness that we call a biofilm. And then those organisms that live in that biofilm can actually sort of work together to communicate and then let each other know how happy they are in the biofilm or not, and then adapt to that biofilm based on chemical signals that they give up. And this can actually trigger them to kind of communicate with each other to have certain like gene products that they make and stuff like that as a response to their environment that is surrounding them. And so that's the idea of a biofilm and the role that capsules and slime layers can play in that. But again, having that sticky coating can help you evade the white blood cells of the host, which is the immune response, preventing phagocytosis. Just like if you were coated in a thick, thick layer of honey, it would be hard to grab onto you if you were running away. So how do these guys run away? Well, they are motile because of flagella. Not all bacteria are motile, but if they are motile, they have flagella. And there are different arrangements of flagella that we can see. Again, if you have flagella, then we would say you are trichous. Trichous comes from the word for hair, like the Latin word for hair, trick, okay? So they're hair-like projections on the outside of the cell. If you are a cell that has just one, then we would say you are monotrichous. If you are a cell that has a tuft or a bunch like that, then we would say you are lophotrichous. If you are a cell that is absolutely just covered in flagella, then we would say you are peritrichous. Okay. And if you are a cell that has at each end, again, that would be both, which is amphitrichous. If you do not have flagella, you don't fall into this category. A lot of our cocci type of bacteria, like staphylococci, for example, a lot of them are non-motile, so they lack flagella. But any of our bacteria that are motile, they have at least one flagellum. Okay, so flagellum is the singular for that, and that's the gist of how that will work. Dismiss. Okay. So what else do I want to say about this? Am I recording this? Please tell me I'm recording this. Yes, okay. I got worried for a second there. Okay. So this is the gist of how the flagella will work and how they actually work to move the cell around. Even if you just have one, each flagella can be moved individually. And so they are just like protein extensions that come off of the cell, and they rotate either clockwise or counterclockwise. And based on that rotation, will allow the cell to move. Now, the cell will rotate the flagella, and that allows the cell to do a run. And that's just a continuous movement. And then they'll suddenly rotate it the other way. The flagellum will rotate the other way, and that'll cause the cell to actually tumble. And then it's just like a coordination of running and tumbling that allows the cell to create movement. And so it's not super controlled, but in generally speaking, if we just run a little bit and then tumble, they'll coordinate that basically to drive what direction they're moving in. The more flagella you have, the more direction you might be able to get by moving this flagella or that flagella, and they can be individually controlled. So that allows for a little bit of directed movement. But flagella do not move in a whip-like fashion. They just rotate one way or another. And so that's what generates motility for our cells. All right. Next, we talk about our cell envelope. The cell envelope is a term that refers to all of the outside levels of the cell. The cell envelope includes the cytoplasmic membrane. All cells have a cytoplasmic membrane. All cells. All cells have a cytoplasmic membrane, okay? Eukaryote and bacteria and archaea, okay? For bacteria and eukaryotes, that plasma membrane is made up of that lipid bilayer, the phospholipid heads on the outside with their little tails that are hydrophobic on the inside. And again, this would just be a continuous layer. And that this is what I mean when I draw that blue line in those cells. Okay, so this is our phospholipid bilayer, and this just continues and covers the whole cell. So bacteria and eukaryotes have a phospholipid bilayer. Archaea have a weird hydrocarbon structure that you don't need to worry about, but it's not phospholipids, okay? So it's just weirder. It's not the same structure. So all cells have a cytoplasmic membrane. Bacteria and eukaryotes have a phospholipid bilayer membrane that contains the cytoplasm and everything in it, okay? Then some bacteria have a wall. Most bacteria will have a wall. That wall could be thick, which is in our gram-positives, or thin in our gram-negatives. Our gram-negatives also have an outer membrane, just for extra stability and control of what goes in and out of the cell. And then we have deviations from these. Okay, so our gram-positives could have a special wax on their outside, which is called mycolic acid. Not all gram-positives have this. So only the genera of Nocardia and Mycobacterium have mycolic acid wax. Those are gram-positive. And again, the genera would be in the name. And the only one I'm going to test you on is Mycobacterium because it's medically relevant. And so this would be like Mycobacterium tuberculosis, Mycobacterium lepracy, Mycobacterium avium complex, which causes tuberculosis disease in immunocompromised folks, okay? So these are relevant. And they have that special wax on their gram-positive wall. So it's still a gram-positive organism, but they also have this wax, mycolic acid wax, okay? And that makes them resistant to being like dried out and resistant to cleaners and things like that, okay? Gram-negatives, there are some gram-negatives that have no cell wall. They just have the two outer membranes, and they have sterols to help stabilize it a little bit. They also kind of are pleomorphic because of that. Again, this is a type of gram-negative that has no wall. Pleomorphic, again, meaning they don't have a set defined shape. They can kind of have a variety of shapes. And this is a genera, a genus, actually. Mycoplasma, which I remember because if I had an actual cup of your plasma that you donated at CSL or something like that, plasma is a liquid. If I pour your plasma out on the table, will it hold its shape? No, neither do these cells. The mycoplasmas do not hold their shape because they don't have a cell wall. The mycobacteriums are like regular bacteria. They have a cell wall that has mycolic acid wax in it, so they're extra, okay? So that's how I remember which one is which. Whatever works for you, though, but they are not related. They are completely different. So don't confuse them just because they have myco in their names, okay? They are medically relevant as well. The mycoplasmas are associated with walking pneumonia. It is not the most common type of pneumonia, by the way. The most common type of pneumonia is caused by Streptococcus pneumoniae, which we will learn about in future chapters. You don't need to know that for the exam yet, but you will need to know it in future chapters. I'm just letting you know so that we're keeping reference. Mycoplasma pneumoniae causes walking pneumonia. So if you have fluid in your lungs, but you're otherwise able to function, it's usually caused by Mycoplasma pneumoniae. Again, they don't have a cell wall, which is a unique characteristic that puts them as a type of gram-negative. When we're trying to learn about whether or not our organism is gram-positive or gram-negative, again, if they have a thick wall versus having a thin wall and an outer membrane, these are huge defining characteristics that basically determine how they can behave in certain environments and what kind of diseases they can live in and what part of your body they can survive in and how they're adapted for things and all that. So it's useful for us to distinguish them based on those cell walls and things like that. So this comes back to the gram stain. Okay. I'm going to get rid of our flagella for a second so we can go over the gram stain. So the gram stain has 4 major steps. The 4 steps of the gram stain, the first step is crystal violet. And this sticks in the peptidoglycan like this. It gets stuck inside of the peptidoglycan. So it would get stuck in our gram positives wall, but it doesn't really stick inside of the wall of the gram negatives. So at first it'll just kind of hang out, just hanging out on the outside of the cell because it is a positive stain. But it'll just hang out there, okay? That's important because we're going to come back to it, okay? Because the next step is iodine, which is a mordant. What is a mordant? It's a fixative. If the crystal violet got stuck in place, the iodine fixes it so that it won't move. This doesn't happen in the gram negatives because it's not really stuck in place, okay? And then our next step is our decolorizer, which is ethanol. It will remove any crystal violet that didn't get stuck in place. So again, what would happen with our gram negatives? It'll just remove what didn't get stuck, but our gram positives will stay crystal violet. But we remove the crystal violet from the gram negatives because it didn't get fixed in place, but not from the gram positives. So the gram positives would stay purple at this point. The gram negatives would be colorless at this point. Okay. And then our final step is our counter stain, which we use saffron in. It is red, just a different color than what we would see with the crystal violet to give some contrast there, okay? So it looks pinkish. It's a faint red, okay? So what this means is we're still staining our gram positives, but the purple is so much more vivid that we don't see the red, but our gram negatives now are not colorless. Now they are red, a faint red, pink, okay? So gram positives stain purple with the gram stain. Gram negatives stain red with the gram stain. We will see this in lab 3. Okay. Which I think everybody's going to get that in. Yes, everybody will have done lab 3 by the time that we get to the Unit 1 exam. So you'll get to see this in action before we get to the Unit 1 exam, which is good. Okay, so that's the gist of that. Okay, so some general structure of that. There's one more subset of our gram positives that I want to talk a little bit about. I'm just cleaning up my screen. And that is our endosporoformers. An endospore is a hardy, resistant, completely dormant, and inert. It's not doing anything except waiting out for better conditions. Some genera of bacteria that can do this, they will have bacillus as part of their first name. So even like lactobacillus, for example, if bacillus is in their first name, the genus, then they can make endospores. Okay. Or if they have Clostridium or a subset of Clostridium, which is Clostridioides. Clostrid. Okay. If their name is Clostrid or has bacillus in it, then they are endospore formers. These are the only ones. Both of them, importantly, are gram positive. So therefore, if you are an endospore former, you are gram positive. So that's something we know about you immediately. If your name is Bacillus anthracis, Bacillus anthracis. We know two things about you immediately. Well, we know three things. Number one, your name is Bacillus. You are rod-shaped, right? We learned about that in our cell shapes. Number two, your name is Bacillus. You're an endospore former. Number three, your name is Bacillus. You are gram-positive. Okay, so all of our endospore formers are gram-positive. Bacillus means you're rod-shaped. And, you're an endospore former. So that's what you know about that guy immediately from its name. Clostridium tetani. Now, we don't know what shape a clostridium is. Not from the name right away. As it turns out, they're club-shaped, okay? But that would fall in the bacillus category. But they're gram-positive. And endospores, just because we know they're endospore former, we know they're gram-positive. So that's something we know about them, okay? Our thick wall guys are gram-positives, and some of our thick wall guys can make endospores. These are hardy, resistant, dormant structures. These are the guys that survive the 99.9% effective cleaners. Okay, very resistant to chemicals, to heat, and radiation, just all sorts of stuff. So they're very resistant. And basically, when you're an endospore form of your cell, you are inert. You're not doing anything else. Endospores do not make toxins. Endospores do not multiply. Endospores do not infect. But you can get an endospore into your body. And then the active form is vegetative. So I want to, yes, just bear with me. I want to kind of draw this out. Okay, so basically vegetative is active. You are multiplying, you are metabolically active, you can make toxins, you can cause infections, and you can grow. Then you can turn into an endospore. You are hardy and resistant, but you are dormant. And you can switch back and forth between them. If you cannot make endospores, if you're not a bacillus or a clostridium, you are Staphylococcus aureus. You are just vegetative. Most bacteria just stay vegetative. But if you can switch into the endospore form and become dormant, then that endospore can survive harsh conditions. The process of forming an endospore is called sporulation. It takes about 6 hours. And then once that endospore gets into favorable conditions, you step on a nail and an endospore gets into your foot. And now we can start becoming vegetative. That process is called germination. And the endospore basically just has a very thick coat that we call a cortex that is protective around it. And it's basically just a thick cortex coat that covers DNA and just keeps it safe. Once we get the right nutrition and stuff, it only takes about 1.5 hours to become vegetative again and start multiplying. But you have to become vegetative to start making the toxins that cause tetanus or whatever. Okay. And I think that is it for that part. And the last part of this was our sorting our eukaryotes based on their cell envelopes. So I'm going to go back to this slide really quick and clean it up really quick. Yes. And so we have 4 categories of eukaryotes based on their cell envelope. Remember what a cell envelope is. It is the cytoplasmic or plasma membrane, the inner membrane, plus whatever cell wall you may or may not have, plus your outer membrane if you do or do not have it. That's your side. That's your cell envelope. So there's 4 categories. The first one that I always remember is the firmicutes. They're firm because they have a thick wall. These are our gram-positives, our traditional gram-positives. Okay. The second one are our other grams, the gracilicutes. These have a thin wall, but they still have a wall. These are our gram-negatives. Our third category are our tenericutes. Tener, like tender. They're so tender because they have no wall. These are mycoplasmas. Remember, like plasma doesn't hold its shape. These guys are pleomorphic. Okay, they have no wall. And then our fourth category are our mendozicutes. These are our archaea. Remember, I told you they don't really have phospholipids. They don't have peptidoglycan. They got other molecules going on. They weird. Those are our archaea. They're in their own category. Okay. So those are our subtypes based on those cell envelopes. So now moving on, we're going to talk about the most missed topics from these chapters. Our first one is from chapter 2. I think we've got one from chapter 2 and the rest are from chapter 4. This is just reminding us about our different types of bonds. We've got our covalent bonds where we are sharing electrons. We've got ionic bonds where we are donating or transferring electrons. And then we have our hydrogen bonds, which are just attractive forces between a polar hydrogen and an electronegative or negatively charged atom. Opposites are attracting. So it's the gist of those. This page goes over it in a little bit more detail. You are welcome to read it if you need extra notes on that stuff. So let's go into our example questions about these topics. Again, our first question is the bond between magnesium and SO4. And I did not balance it, but as it turns out, the magnesium and SO4s, these are 2s that are charging on these guys, but I'm not that worried about it. If you've got an atom that is an actual plus and another part of the molecule that is an actual negative, what happened was an electron from this atom was donated to the other atom, which is why. And in this case, it would actually be 2 electrons if we had 2 charge. And that's what's really happening between those guys. We're actually donating or transferring those electrons. We're not sharing them or anything. And then they dissociate or ionize completely in water. So the bond between magnesium and sulfate allows it to dissociate when dissolved in water because those ions are interacting with the water molecules. What kind of bond is that? Are they sharing their electrons? They are not. That would be covalent. Are they attracted by a positive polar hydrogen? No. Is this van der Waals forces? And those are attractive forces too, but not involving a hydrogen. We don't need to know them for my class, so that's not going to be the answer. This is an ionic bond. We have actually transferred an electron from one molecule to another, in this case, more than one. DNA exists as a double helix. Sorry, double helix, that whenever it's unwound as a ladder. Okay, and these 2 strands of sugar phosphate backbones that each one is bound to a nitrogenous base that is hydrogen bonded to its match on the other side. And then these guys are held together by hydrogen bonds. So DNA exists as a double helix. It is called double because there's 2 strands held together by hydrogen bonds, which we can see there. Okay. In any one of those given bonds, what type of electron interaction is happening? Are we actually having interaction with electrons there? No, we are having attractive forces between polarities, polar attractions. No electron transfers. So our answer is C here. We're not sharing electrons. We're not donating electrons. And negative electrons are not becoming positively charged. That's not what's happening here. Normal chemistry stuff here. Okay. Our next topic is from chapter 4. This deals with gram-positive and gram-negative differences, reminding us that on our gram-positives, we have a thick wall that needs help from like lipoteichoic and teichoic acids. Our gram-negatives have a thin wall that has an outer membrane for a little extra protection and support, and that outer membrane has lipopolysaccharides, lipo being fatty acids, polysaccharides, many sugars. We abbreviate as LPS, and that is also known as endotoxin. It's endo because it's in endo side of the cell, it's part of the cell, and it's toxin because it causes damages to our bodies. Okay. That's the gist of the differences between the gram-positives and the gram-negatives. So going on to our questions about this, again, gram-positives, thick wall. Actually, we need more specific. Thick peptidoglycan wall supported by lipoteichoic and teichoic acids. You don't need to know what these are. You just need to know that they support the wall and maintain the wall, okay? Whereas our gram-negatives have a thin peptidoglycan wall with an outer membrane, which is an extra feature that contains lipopolysaccharide, which is also known as endotoxin. Okay. That's our little cheat sheet note for these questions that we have next. So a patient sample is tested and it found that it has lipoteichoic acid. Which of these two has lipoteichoic acid? Do gram-positives have lipoteichoic acid? Yes, they do. Do gram-negatives have lipoteichoic acid? No, they have lipopolysaccharide. Do acid-fast bacteria have lipoteichoic acid? Now, acid-fasts, these guys are the guys that have mycolic acid wax. They're also known as acid-fast. Okay, it has to do with the staining procedure, which is lab 4. Okay, we won't get to lab 4 before our exam for our Monday, Wednesday people. We should get to it for our Tuesday, Thursday people. But the acid-fast stain basically is a stain that we use a lipid-soluble stain to stain the mycolic acid wax. It is lipid-soluble, and if it's lipid-soluble stain, it'll stick to the wax. It won't stick to cells that don't have the wax. And then we use acid to rinse it out. If it doesn't rinse it out, it's acid-fast. It has mycolic acid wax. If it does rinse it out, the acid rinses it out, it's not acid-fast. Okay, so that's the example. That's the ideology behind that. And if you want more detail on that, it is in lab 4, so you're welcome to look up more detail about that and how that stain works and the concept of how it works if you are interested before you get to the Unit 1 exam. But anyways, the idea is with the acid-fast stain, that's staining for the presence of mycolic acid wax. These are gram-positive organisms, but they're only specific gram-positive, so the mycobacteriums. So this is partially correct, but the A is the best answer here because all gram-positives will have lipoteichoic acid. Okay. And you can tell because this is a feature of our gram-positives. So A is our best answer here. Which of the following is not a feature of our gram-positive outer membrane? Where do we find an outer membrane? The gram-negatives. So gram-negatives have an outer membrane with LPS, but not gram-positives. So none of these shouldn't have an outer membrane. Okay. Our next concept is the endospores. Dealing with the endospores. Yes. So the gist here is that the endospores are essential for survival, but they are inert. Only the vegetative form is the growing form. So we're going to kind of give a little bit of there's the information on that sheet, but I'm going to make our own little cheat sheet before we start getting to the questions. Remember, if you are an endospore, you are inert and dormant, and then you become vegetative. This is the opposite of that I drew before, but it's the same concept, which is the active form that actually multiplies and causes the infection. When you are vegetative becoming an endospore, that is sporulation. If you're an endospore becoming active again, that's germination. Think of the endospore as being like a seed. The seed isn't growing. Once the seed starts to grow, it's becoming a plant, it's becoming vegetation. And that seed, when it is becoming vegetation, is germinating. So it's the same concept here, okay? All right, so a researcher discovers a bacterial strain that forms endospores, but lacks certain genes associated with the coat, that cortex, that thick outer coating. So it's not going to be as hardy, probably. After exposure to heat, the strain shows reduced survival compared to other normal endospores. Okay. Which of the following conclusions can be drawn from this observation? The bacterial strain's reduced survival is because of lack of DNA replication. I don't know how that would be related. Is that possible? Potentially, but is there a better answer than that? The absence of the spore coat genes. So we have, because we can't make a proper spore coat, this makes it so that we, impacts the ability of the endospore to withstand extreme environmental conditions. Because we don't have the spore coat properly, we can't survive extreme environmental conditions like too hot or too dry or whatever. This seems like our best answer so far. The reduction in survival rate suggested that bacterial strains endospores have a higher metabolic activity. If they were higher metabolic activity, they'd be growing better. The strains endospores are likely to be more resistant to chemical disinfectants. More resistant? I'd assume they'd be less resistant. So it's probably not that either. So B is our best answer. A patient of yours is diagnosed with C. diff, Clostridium difficile, Clostridioides difficile. Okay, so this is an endospore former. On exam questions, I will give you the full name. I would say Clostridium difficile. Okay. Why would containment protocol be more strict for Clostridium difficile than for Salmonella? Because Clostridium difficile is an endospore former, and endospores are hardy and can resist the cleaners, right? Salmonella, does it form endospores? No, because it's not Bacillus or Clostridium in its name. So Salmonella is endospore forming? No. Clostridium is. Clostridium difficile is endospore forming? Yes, making it hardy and resistant to cleaning and cleaning methods. All gram-negative bacteria are endospore forming. This is incorrect. Only certain gram-positives are. Not even all gram-positives can. All diarrhea-causing bacteria are endospore forming. Obviously not, because you know that Salmonella causes diarrhea, E. coli causes diarrhea, and there's other ones too, trust me, that we'll learn about. Not all of them are Bacillus or Clostridium. Okay, so B is our best answer here. All right, so our next one, this is our last concept, is differentiating by that cell envelope. And so this is talking about the firmicutes, gracilicutes, mendozicutes, and the tenericutes there, basically based on that cell envelope. And remember, the cell envelope is the plasma inner membrane, whether you have a wall or not, whether you have an outer membrane, and what's your membrane made out of. Okay, so we'll have our little notes out to the side on this one again. Whoops. We'll have our little notes out. Trying. Okay, so on this one, we have gram-positives are firmicutes. Gram-negatives are gracilicutes. No wall is tenericutes. And then Archaea are mendozicutes. So those are a little cheat sheet to refer to as we answer these questions. A microbiologist is studying a bacterial strain that has a thick peptidoglycan layer that sounds like gram-positive in its cell wall. Based on this description, which group does it belong to? Well, it's a gram-positive. Which group do the gram-positives belong to? The firmicutes. The gracilicutes are gram-negative. The mendozicutes are Archaea. And the tenericutes are the no wall, which is really just mostly our mycoplasmas. All right. 56-year-old patient presents to the hospital with severe GI symptoms, including nausea, vomiting, and abdominal pain. Laboratory tests revealed that the causative agent is gram-positive. We got a thick wall. It's rod-shaped bacteria, so it's a bacillus shape. Okay. Upon further testing, it's noted that bacteria's wall is thick. Again, gram-positive, thick wall, that tracks. And it shows resistance to desiccation and high temperatures. That suggests that it might be an endospore former. Which category do our endospore formers fall into? Well, we already know that it's gram-positive because it gave it to us. But if all I gave you was that it was an endospore former, you would be able to recall that the endospores are all gram-positive, so they would be falling into the firmicutes. Remember, gracilicutes are gram-negative, and they're not endospore formers. Mendosicutes are no wall, which is a subset kind of the gram-negatives. But specifically, no wall is mendosicutes. Those are mycoplasmas, not endospore formers. The tenericutes, oh, I got those wrong. The tenericutes are our no wall. I got those switched. And the mendosicutes are Archaea. And gracilicutes are gram-negative. None of these make endospores. Only gram-positives can make endospores. So even if I just gave you that, that would be enough information to answer the question, but I gave you a lot more. All right, so that is the gist of the content for chapter 2 and 4 for week 3's content. So if you guys have any questions, please reach out to me. And while you're studying, keep in mind that your, because we had a question about this in our discussion area, keep in mind that your learning objectives are your guidelines for what you need to get from the chapter. And so answer your learning objectives with what information you get from the lecture. The lecture may go into extra detail about some things that might give context, but it doesn't always mean that you have to know all of that information in that lecture. So use the lecture information to answer your learning objectives at a level that you're answering the learning objectives, but not beyond them, but well enough that you could have discussions about the content. And that's the point of your lectures. And there's a lot of repetition here. I'm aware of that, but that's on purpose to help drill a lot of these concepts into your brain. So reach out to me if you have any concerns about any of that or if you're having issues. And remember that the SSOC and the STEM lab are there as resources for you guys studying as well. And I hope to see you again soon. Thanks.